the boundary of our sarcomere. But within that, we have this M line, which is another type of protein. And then we have this guy here that's coming from that M line. This is a protein called myosin. These proteins here that are coming from this M line, that's called M line, right? Which being anchored, that M line is anchoring these proteins called myosin. Right? Our Z disc is an anchor that's anchoring which proteins? Actin. So what we have here, these proteins are called actin. And what we call our actin and myosin, both of those are considered myofilaments. Those are called myofilaments. Our myosin is our thick myofilament. But our actin is our what? Thin. So what our sarcomere contains is thin myofilaments and thick myofilaments. Again, our thick myofilament is called who? Very good. And our thin myofilament is called what? And it's going to be a relationship between the two of our friends that actually create the tension, which is contractility. It's the myosin and actin relationship that's going to create contractility. Right. By definition, again, what is contractility? When the actual muscle generates tension. Right. So now what we're going to see now is this. What we're going to see is that our myosin has arms. Myosin has arms. Okay? All right? Those arms are called, okay? And really, I said arms. They're considered myosin heads, okay? But I like that. When I show what I'm going to show, I like to look at it as these arms. All right? These arms that have hands. But they're called myosin heads. Okay? And so, what the intention of these arms to do, what their intention is to reach out. See, these are the arms of the myosin. See that? They're reaching out, and their intention is to attach to what? Actin. Okay? That's what they want to do. Reach out and attach. Okay? Reach out and attach. Now, if they do so, then they will make that attachment to actin. And then they're going to do what? Pull. They're going to pull on the actin. They're going to pull. When they pull on the actin, okay? When they pull on this actin, how are they pulling? They're pulling it toward the what line? There you go. Okay. So when these actual myosin arms, and when their hands attach to the actin, it pulls on the actin. And what is the actin anchored to? The Z disc. And that's what brings the Z disc closer to the what line? Okay, you see that? What you're seeing is that when you have your myosin attached to the actin and then pull on the actin is going to pull who? The Z disc. Z disc come what? Closer to yeah. Okay? Yes. All right? So what you're going to have now is this same guy, once those myosin arms pull, what we're going to have is these same myosin filaments, but then now we're going to have Z discs that are what? Closer to each other. You see that D disc is now closer here That Z disc is what? Closer there. And we even have our actin filaments closer to the what? Closer to the M line. Okay? And as a result of this pulling, we have a sarcomere that's what? Yeah. It's much shorter. We have a sarcomere in length that's much what? Shorter. What does that do now? What happens to one sarcomere happens to what? All the successive sarcomeres. You see that? And now what's going to have to happen? Tension is actually going to be generated, yes? On the what? Sarcolemma. And now that makes the whole what? Muscle cell what? Shorter. Isn't that beautiful? But our actual sarcolemma does not what? Cave in and actually break apart, right? Because it's only oil consistency. It will be able to take that tension because what? What protein is there to shield it? Dystrophin. Dystrophin will be able to make sure that this plasma membrane can take that tension and not fall apart. Okay? Very good. Okay? So that's what we're looking at. All right? And what I just described, you have a question there?
What did you call the little arm that
Very good. So the arms are really actually considered what we call myosin, because where do they come from? The myosin. They come from the myosin myofilament. So they're called myosin heads. They're called myosin heads. I know I said arms with hands because when I want to demonstrate this process, I want you to see it in that way first, but we're still going to consider the myosin heads at the end here. All right? So this is how it works, okay? Yeah, Chelsea, I'll need you as a model. China, a model as well. I need models, okay? Chelsea, get up. And China, get up. I want you. Now, you guys are pretty tall. So they're going to be our actin filaments, all right? And you guys are going to face each other, okay? All right? Very good. All right. I need you. Yeah. And we're going to be the actual M line. China, you guys are tall. It makes me feel bad. All right. So now we're the M line. So what she's going to do is face China, right? Okay. And my back. So my back and her back is the what? M line. Okay. Yeah. So now what we're going to do is she's going to reach out her arm. Now M line anchors the what filament? The myosin. Okay, very good. And my arm, mine sticking out also represents what? The myosin. Okay. All right, now they're going to stick their arms out. Right? Go ahead. And now that means that her arm represents what? And then Chelsea, do that deltoid. See, it's an exercise. See? If she keeps it up there, she's what? She's generating tension. What is that called? Contractility. So basically, what is her arm? China's arm is what again? The actin. And Chelsea's is what? Actin. There we go. All right, so we get it. So I want to represent that this myosin head has these particular what? Heads, but we're going to represent them as hands right now. And say that they reach out, and their purpose is to grab the what? Actin. Is that right? So she's going to grab, there we go. Myosin heads, what? Grab the actin. And then they do what? They pull. And now you can see that the what? Actin gets closer to the what line? The M line. And that actin gets closer to the M line. And as a result, what happened? The Z disc came what? Closer. Beautiful. Now, I want to add this. Keep your arm down for right now. Arms down. We're just going to focus on Chelsea and I. And I want you to look at half the sarcomere. And so what the actual what? This is what her actin has on it. It's another protein. Okay? Actin has this protein on it called tropomyosin. I'll write the name up in a second. Tropomyosin is special because what it's blocking is this. It's blocking the place where I would ordinarily do what? where I would bind. The myosin head has to bind to the actin, but this protein's covering it. Okay. What's this protein called? He's stopping the process of the myosin head binding. All right. So this is the special thing, though, right? All right. This protein as well is attached to the tropomyosin. What's this protein called? Troponin. I'm gonna write these names down in a second. Let's keep these two proteins in mind. They're involved with the process because tropomyosin is blocking the place where the myosin head is supposed to attach to, and troponin is attached to the tropomyosin. Let's consider the tropomyosin then a sheet that's covering the place that the myosin head has to bind. And now we're going to consider that this troponin is just stuck to this what? So this is what happens, right? When you actually have calcium go into the muscle cell and amongst all of these myofibrils and sarcomere, calcium binds to this troponin that's stuck to the tropomyosin. And when calcium binds to the troponin, it changes the shape of him so that now, much heavier, he then what? Comes off of this actual what? Myosin myofilament, right? So when troponin comes off, what is he stuck to? He's stuck to what? Tropomyosin. And then what happens is that when calcium hits troponin, changes troponin to the point where he falls off, now troponin pulls who off as well? And now who's exposed? The actin, so that the myosin can do what? Bind to it. Now I can bind, and now I can do what? Pull. I cannot pull unless what happens? Calcium binds to who? Troponin. Troponin goes through his chain, falls off, but then pulls who with him? Now you can have the actual place where myosin head can bind to the what? Actin. You will not have this happen unless calcium's involved. You got that? All right, sit down. Thank you so much. So we will not have this process occur unless calcium's involved. And what's going to happen is this. All right? So what do we have then? We have a situation where myosin heads have to attach. One element's going to be responsible for that. But then we also have to have calcium involved to make the actual place where myosin binds available. Calcium needs to be involved, and then we're going to have our actual myosin head attached. All right, now, let's back up for a second. All right. Let's back up. What I explained to you when the myosin head binds to actin is the process of contraction. Isn't that right? But now let's make this clear. That process doesn't just happen out of the blue. Myosin can't just move like that. It's a protein. It will not just move mechanically like that, out of the blue spontaneously, okay? Whenever something moves, what do you have to put into it? Energy. Whenever you're working, you got to put what into it? Energy, okay? So now, let's go back to that major energy currency in our cell. And what is that? What's our energy currency for? So anything that happens, whenever you need energy, whenever the cell needs energy for a whole bunch of chemical reactions to occur, you have to have who there? ATP. The way that the myosin head is going to actually move and make his attachment, the only way he's going to move is by having energy. And how is he going to get that energy? By which type of substance? Got it? Hence, the only way that you're going to start contraction, which is myosin head binding to actin, is by having who? ATP. The main thing needed so that myosin moves